So in this video, we will be discussing about an important topic in pathology, which is cell injury. So we'll discuss on the causes of cell injury, the response of the cell to the injury, and we'll see the ultrastructural changes or electron microscopic changes of the cell in a cell injury. At last, we'll discuss a few MCQs on this topic, which have been asked in various exams. So first, we'll discuss about the causes of cell injury. So causes of cell injury. First of all, so any disease involves a functional alteration in the cellular level. So we know that a cell is being a tissue is being supplied by blood vessel. So inside this blood vessel, the hemoglobin carries the oxygen with it. So when there is any decreased oxygen supply to the cell due to any cause, that is called hypoxia. So hypoxia is a condition in which the cell doesn't get enough oxygen. So there is deficiency of oxygen to the cell. So hypoxia is the most common cause of cell injury. Most common cause of cell injury. And we know that so the decreased oxygen supply to the cell can be due to decreased blood supply. So when the blood supply to the organ is decreased, or it may be because of decreased hemoglobin in the blood to carry the oxygen, that is due to anemia, or it may be due to decreased oxygenation of the blood due to lung disease or heart disease. So which all causes can lead to hypoxia? One is decreased blood supply. Hypoxia due to decreased blood supply is called ischemia, and decreased hemoglobin, that is anemia, or decreased oxygen carrying capacity of hemoglobin due to carbon monoxide poisoning can also occur, or decreased oxygenation due to lung disease or heart disease. Due to lung disease or heart disease. Lung or heart disease. So out of this, hypoxia due to ischemia is the most common cause. Out of this, ischemia is the most common cause. So what are the other causes of cell injury? It can be due to some physical agents like thermal injury. It can be due to some burns, or it can be due to irradiation, can be due to pressure, frostbite, like that, and can be due to some chemical agents like poisons, like acids and alkali, can be due to some drugs also. Some immunological causes, immunological phenomena which can lead to cell injury are autoimmune reactions. Autoimmunity, which damages the cells, can be due to hypersensitivity. Can be due to anaphylactic reactions, anaphylaxis. So these are the immunological causes, or it can be due to some nutritional causes, nutritional deficiency. It's nutritional causes, or aging can also lead to cell injury. Some idiopathic causes and idiopathic causes. Iatrogenic causes can also result in cell injury. So these are the causes of cell injury. Out of this, the most common cause of cell injury is hypoxia. In hypoxia due to decreased blood supply to the tissue is the most common cause. So ischemia is the most common cause. Now we will see the response of a cell to cell injury. Response to cell injury. So, a cell can respond to a cell injury in three ways. First of all, the cell can adapt. So, adaptation, that is, it will alter its physiological function in response to cell injury, that is called adaptation. Or the cell can get reversibly injured, that is called reversible injury, in which the cell is able to regain its function if the cause of cell injury is removed. Reversible injury. Or the cell can get irreversibly injured, that is called irreversible injury. Irreversible injury to the cell can lead to necrosis or death of the cell. So these are the responses of the cell to cell injury. Now we will see reversible cell injury and the changes which take place in reversible cell injury. So just imagine a cell here. So there is hypoxia, that is hypoxia, decreased oxygen supply to the cell. So decreased oxygen supply to the cell will lead to what happens? There is an organelle here, that is mitochondria. So because of the decreased oxygen supply, what happens? The mitochondria dysfunction occurs. Mitochondrial dysfunction. Mitochondrial dysfunction occur. So because the oxidative phosphorylation in mitochondria needs oxygen. So oxidative phosphorylation doesn't take place and decreased ATP synthesis occurs. Decreased ATP. So all other changes are subsequent to this decreased ATP synthesis. We know that ATP is needed for the sodium potassium pump to work effectively. So the sodium potassium pump normally pumps out sodium and pumps in potassium. So if ATP is decreased, the sodium potassium pump will not work and inside the cell sodium accumulates. So when sodium accumulates in the cell, it will lead to accumulation of water also to maintain the isosmotic condition. So accumulation of water, H2O also. This will lead to hydropic changes in the cell. Hydropic changes, that is, cell becomes swelled up. Swelling of cell occurs. Swelling of the cell occurs along with the organelle. The cell goes into a stage of hydropic change. And other changes are because of the decreased ATP and decreased blood supply, there will be increased anaerobic glycolysis. Anaerobic glycolysis take place. So we know that the product of anaerobic glycolysis is lactic acid. So when this lactic acid increases, the pH of the cell will decrease. pH decreases. When the pH decreases or it becomes acidic, there will be clumping of nuclear chromatin. Clumping of nuclear chromatin. And because of increased glycolysis or anaerobic glycolysis, there will be increased glycogenolysis. So another change which occur here is because of the decreased synthesis of ATP and because of a increased hydropic change inside the cell, the ribosomes will get the ribosomes will get detached from the rough endoplasmic reticulum. Ribosomes gets detached from rough endoplasmic reticulum, and this leads to decreased protein synthesis also. Decreased protein synthesis. So because of this hydrophic change, what will happen? The cell will get swelling, organelle get swelled. So because of the swelling of the cell with water, the cytoplasm of the cell will be forming some blebs cytoplasmic blebs to accommodate more water. So because of the hydrophic change, there will be loss of microvilli also. Loss of microvilli. Now, the changes are changes in reversible cell injury are let's see that. So what are the cytoplasmic changes we see here? So because of the increased accumulation of water inside the cell as such gets swelled up. That is called cell swelling occur. That is first change. That is also called hydrophic change. So hydrophic change is the earliest change seen in cell injury. Earliest change in cell injury is hydrophic change. So now what happens? Because of the increased accumulation of water inside the cell, the cytoplasm will form blebs to accommodate more water. So that is called cytoplasmic bleb. This is called cytoplasmic bleb. And this is loss of microvilli. Because of the water accumulation, there will be loss of microvilli. And we have seen that the rough endoplasmic reticulum that also gets swelled up. So swelling of rough endoplasmic reticulum RER with detachment of ribosomes from the rough endoplasmic reticulum. Detachment of ribosomes. Now we will see another structure which is present here, that is myelin figures. So myelin figures are a characteristic feature which is seen in reversible cell injury myelin figures. So these are actually the phospholipids that are formed from the damage to the cell membrane and the cell membrane of the organelles, which contains water and lipoproteins accumulated in it. So the phospholipids, myelin figures are phospholipids with accumulated water and lipoproteins. Accumulated water and lipoproteins. So myelin figures are also seen. And next, the lysosomes will exhibit a phenomenon which is called autophagy. So it will start phagocytosing its own organelle. Autophagy of lysosomes. And the cytoskeleton forms aggregates. So these are the changes. And what is the change in nucleus? We are saying that because of the anaerobic glycolysis, lactic acid is formed, which leads to decreased pH. Due to decreased pH, the nuclear chromatin clumps are formed. Nuclear chromatin clumps are formed. So remember the changes in these are actually ultra-structural changes in reversible cell injury. Ultra-structural changes. So cell swelling or hydropy change, that is the earliest change. Second one, cytoplasmic blood formation, loss of microvilli, swelling of rough endoplasmic reticulum with the detachment of ribosomes leading to decreased protein synthesis. Myelin figures, which are actually phospholipids with accumulated water and lipoproteins. Lysosomes exhibiting autophagy in the cytoskeleton aggregates and clumping of nuclear chromatin. These are the features of ultrastructural features of reversible cell injury. Now we will discuss the changes, ultrastructural changes in irreversible cell injury. Ultrastructural changes in irreversible injury. So the characteristic feature of irreversible cell injury is that there is mitochondrial dysfunction. Mitochondrial dysfunction which is not improving even with restoration of blood flow or reperfusion. Not improving with reperfusion, and continuing cell membrane damage. Membrane damage is continuing. These are the features of irreversible cell injury. Membrane damage with mitochondrial dysfunction, not improving with reperfusion. So the characteristic features. Now what happens in irreversible cell injury? We have seen that the cell membrane is already being damaged, so phospholipid synthesis does not occur due to decreased ATP synthesis. So cell membrane is damaged, and because of the damage there is an increased permeability to calcium, and calcium influx to the cell occurs. That is, calcium influx occurs. So when this calcium influx occurs, the calcium is toxic to the mitochondria again. So the calcium salts accumulates in the inner mitochondrial membrane, forming amorphous dense deposit. So these amorphous dense deposits are a characteristic feature of irreversible cell injury. And you know that this also calcium leads to activation of phospholipases. So when this endogenous phospholipases are activated by calcium. So when the endogenous phospholipases are activated, it leads to enzymatic digestion of phospholipids. Phospholipids of the membrane are digested. So, and this leads to membrane damage. Membrane damage. So cell membrane damage. Next, which other enzymes are activated by calcium? It activates proteases. Proteases are activated, and this proteases lead to protein digestion. Proteins are degraded, are digested, and enzymatically degraded, and this leads to cytoskeletal damage. So, cytoskeletal damage. So the cytoskeleton actually anchors the cell membrane. So when the cytoskeleton is damaged, this again indirectly leads to cell membrane damage. Cell membrane damage. So the calcium again activates another enzyme, which is endonucleases. Endonucleases, which digests the nucleic acid and leads to nuclear changes. What are the nuclear changes seen? That is pycnosis. Pycnosis is nuclear condensation. Pycnosis, which is nuclear condensation. And the other change is karyohexis. Karyohexis. Karyohexis means splitting of nucleus. And karyolysis. Karyolysis is actually dissolution of nucleus. Dissolution of nucleus. So the changes which are seen in irreversible cell injury are, because of the calcium influx, there will be amorphous dense deposit in the mitochondria, and because of phospholipases, the membrane, cell membrane and all the organelle membrane are damaged. Because of proteases, cell membrane damage again occurs. Because of the endonucleases, there will be pycnosis, karyohexis and karyolysis. So these are the changes which can be demonstrated like this. If this is a normal cell, what happens here is that the cell membrane is disrupted. Cell membrane is disrupted. And what about the mitochondria? So this is mitochondrial membranes are damaged, and the mitochondria will show amorphous dense deposits because of calcium influx. This is called amorphous dense deposits. So these are the characteristic feature of amorphous dense deposit is a characteristic feature of irreversible cell injury. Then what happens to the other organelle? That is the ribosomes are detached and the rough endoplasmic reticulum. Ribosomes are detached from the rough endoplasmic reticulum, and endoplasmic reticulum is also damaged and disrupted. The structure is again lost. Membranes not disrupted. And what happens to the lysosomes? The lysosomes are broken down. Lysosomes break the cytoskeleton is also damaged. Cytoskeleton is also disrupted. And the nucleus undergoes certain changes. The nucleus may undergo condensation. Nucleus can undergo three changes. It can be epignosis, which is nuclear condensation, karyorrhexis, karyolysis, which is the solution of nucleus. The nucleus can undergo condensation, it can split up. So this is a condensation, it can split up or can get dissolved, dissolution of nucleus. So these are the ultrastructural changes. These are the ultrastructural changes in irreversible cell injury. Ultrastructural changes in irreversible cell injury. That is, the cell membrane is disrupted. There are amorphous dense deposits in the mitochondria. Ribosomes are detached and endoplasmic reticulum is completely damaged and disrupted. Lysosomes will break and release the hydrolytic enzymes into the cell, and this will cause a release of certain enzymes into the blood also. And there are cytoskeletal damage and pignosis, karyorrhexis and karyolysis of the nucleus. So now you can see this is the histology of the myocardium. You can see these are the normal myocardial cells, and this region we can see cells which has undergone swelling here. Cells has undergone swelling here. Vacuolation can be seen. So this is called hydropic change. Hydropic change. So this is hydropic change, which is the earliest change which is seen in cell injury. Now we'll discuss few questions from this section. Is question number 1. All of the following statements are true regarding reversible cell injury except first one, formation of amorphous dense densities in mitochondrial matrix. So this is a change which is seen in irreversible cell injury. And second one, diminished generation of ATP. So that is definitely seen in reversible cell injury. Option C, formation of blebs. That is also seen in reversible cell injury. Option D, detachment of ribosomes from granular endoplasmic reticulum. That is definitely seen in reversible cell injury. So only change which is not seen in reversible cell injury is option A. That is formation of amorphous dense deposits in mitochondrial matrix. So option A is the answer. Second question, ultrastructural finding of irreversible injury. So ribosomal detachment is seen in reversible injury. Second option, amorphous densities. That is an ultrastructural finding in irreversible injury. That is the answer. And question number 3. Question number 3 is, most pathognomonic sign of irreversible cell injury. That is option A, amorphous densities in mitochondria. So option A is the correct answer. Question number 4, irreversible injury in cell is, first one, deposition of calcium in mitochondria. So that is definitely same. And question number 5. First cellular change in hypoxia is, we know that in hypoxia the first cell organelle to get damaged is mitochondria. So that leads to decreased oxidative phosphorylation. So option A is the correct answer. Thank you for seeing this video. To see more videos from our channel, please subscribe our channel. Thank you.